Since the beginning, the human body has been one of the most fascinating realms of science. From muscles and bones, chromosomes and DNA, all the way down to the atoms that make up our bodies, science has been trying to solve the puzzle that we call human. Traditional microscopes help scientists to discover a smaller world within ourselves, but they are limited by diffraction, bending of light around an object that causes the light to disappear when the object is very small. One relatively new technology that helps get around this limitation is called an atomic force microscope. Consisting of a sharp point, robotic arm, and laser, the AFM works by scanning the top of a sample and measuring the deflected motion of the cantilever. The deflections are then put into a computer to construct an image. Such images, like DNA shown here, have given scientists further insights into the molecular world. There are, however, limitations. Molecules on the atomic scale have a tendency to jitter around. Like when taking a photo without a tripod, the image is often blurry. Dr. Gavin King, a researcher at the University of Missouri, has developed a custom instrument with a built-in tripod, so to speak. Only the second ever built, the ultra-stable atomic force microscope, is actively stabilized through a collection of lasers, optics, and computer software. Tina, a grad student in the King lab, is performing sample preparation for a test run of the instrument. First, she puts oil in the lens. This functions to keep the lasers from bending too much as they go through the lens and reflect off of the sample stage. She has to make sure to be delicate, because the black PI stages are the most expensive and important parts of the instrument. Their precision movements are what make the machine so accurate. Gloves are necessary as not to put fingerprints on the sample stage or the oil immersion lens. She has to be careful when raising the lens to the sample stage. Too much, and she risks damaging the stage and the lens. Too little, and the image will not be in focus. As we can see, she knows to stop when the oil spreads evenly on the backside of the sample stage. A fiber optic light is used to illuminate the stage so that the camera can show her a highly magnified image. Here we see 800 nanometer beads. To give you an idea, the length of the entire screen is about the width of a human hair. Now we are ready to see the instrument in action. Here we see the ultra-stable in action. The stage, which can be seen on the left, moves while the tip is kept stationary. The raster pattern, which is on the right, is then used to construct an image. The King Lab's main focus is membrane proteins, the gatekeepers of the cell. In studying membrane proteins, the King Lab hopes to gain valuable insights into the dynamics of molecules. Insights like how drug binding occurs in the cell and how the process might be improved. This research could one day lead to better pharmaceuticals with fewer side effects.